Welcome to the Biology class. Our discussion will be on excretory system today. The excretory system consists of so many things because our kidney is the first. Kidney is related to excretion. Skin excretes something. Then GI tract, lungs. These are the excretory organs. Mainly it is concerned with the kidney. We can find our discussion on kidney today. Now, we all know that kidney excretes urine and it consists of this and that. But firstly, we should understand how it is supplied with the blood. In the kidney, the blood goes and thereafter it excretes urine. So, this is the main artery which supplies blood. Now, this is left ventricular output. So, this left ventricular output, it's 25% of this left ventricular output goes to the kidney through the renal artery. How? Suppose this is the kidney. The renal artery brings the blood and will goes to the kidney. Now, the 25% of this left ventricular output, let us suppose that our blood is 4 liters. So 25% is 1 liter. So 1 liter blood goes to 2 kidneys. So this kidney contains 500 ml, this contains 500 ml per minute. So per minute it is supplied 1 liter of blood is supplied to the kidney per minute. That is, throughout the body, whatever excretory products are being produced, that excretory products goes throughout the body, and out of this ventricular output, 25% goes here for filtration, because kidney is the filtering unit and it filters the urine. This comes through the urinary bladder and then urethra. This much we know. So first we should know that how much percentage of blood goes to kidney per minute. It is 25% of the left ventricular output goes to renal artery and it is supplied to the 2 kidneys in our body. Number 1. Kidney's functions are three types. It has got excretory function. It has got homeostatic function, and kidney is also does the endocrine function also. Kidney secretes some hormones. So kidney performs the excretory, homeostatic and endocrine functions. We will discuss on excretory functions, and although we will discuss these two also a little bit. Now this excretory function done by the kidney. We know that we have two kidneys. So kidneys are like this. It is outer side is convex and this side is concave. This has a special notch. This area is known as hilum. We know that this area is known as hilum. And now it is supplied by artery. So this is known as renal so it gets renal artery goes, supplies the blood and it goes into the kidney, filters there. Now, whatever is produced, that is urine, the urine comes out from here. It's called ureter. This area is known as pelvis. So, it is coming out. Only urine is coming out. Blood is going in. So blood should come out. So renal vein, let us make it in this way. Renal vein goes out. So it is through this hilum or that notch or that concave area through which renal artery gets in, and the two things, that is renal vein and pelvis or ureter, comes out. So it is the single opening here. Now this kidney in length, it is 10 to 12 centimeter. 10 to 12 centimeter is like this, up to this, 12 centimeter. We are making it a bigger one. So it is lengthwise, it is 10 to 12 centimeter, and the breadth is 5 to 6 centimeter. While the thickness is 3 to 4 centimeter in thickness. That is, it is 10 to 12 centimeter is like this, 12 centimeter, and this width is this 5 to 6 centimeter, and the thickness this thickness is to 3 to 4 centimeter. That is two pear-shaped organ. It is situated where? In the abdominal cavity. Now where? It is behind the vertebral column, not here. It is in the back side. That is behind the vertebral column. There are two kidneys on both the side, and it has 10 to 12 centimeter, 5 to 6 centimeter and 3 to centimeter length, width and thickness. Now, it weighs about only 150 grams, and it is performing the main excretory functions of our body. That is 150 grams of a little mass. It is doing the entire work. And the thickness is only 3 to 4 centimeter. It is like this. So within this small organ, the entire excretory product, main excretory product, urinary system is takes place. Now if I cut the kidney, if I does a section from longitudinal section, LS, from this side, if we cut it, it will look like I am making it a bigger one. This is the notch from where pelvis annulator, renal artery, renal vein comes out. Okay. So it is the three things. One goes and two things comes out. Renal vein and ureter comes out. And now it is covered by thick fibrous white tissue. It is in the back side of our body, on both sides of the vertebral column, behind the vertebral column. That is, it is like this. So it should remain suspended or it should remain attached with something. So we should know that these two kidneys remain attached with the peritoneum. It remain attached behind the vertebral column in the abdominal cavity. It remain attached with the peritoneum, and the peritoneum layer in which these two kidneys are attached is known as mesorchium. These two kidneys remain attached behind the vertebral column with peritoneum. The special peritoneum is known as mesorchium. So you should write down that it remains attached with mesorchium. Now, if we section the kidney, cut the kidney from this side, we'll see a deep brown color. This place is known as cortex, and this place is known as medulla. Medulla, this is known as cortex. Now this cortex is divided into three. 1, 2, 3. This is a portion. This is a portion of cortex. It is brown in color, reddish brown in color. I am making medulla a bigger one, although it is 3 to 4 centimeter, 3 to 4 centimeter. But still I am writing in this way. This is medulla. Medulla is divided by pyramid like structure. This is a pyramid. There are 10 to 15 pyramids within that 3 to 4 centimeter area. So these are all pyramid like structure. This is darker red brown and this is lighter, and it has 10 to 15 conical areas. This broad base of this pyramid is towards the cortex, and the shorter one or apex or papilla, the shorter one is towards the hilum. In broad base here and short base or the apex is here. These pyramids are called renal pyramids. Now, here the lateral areas, these lateral areas, are like medullary rays we have gone through in our classes. Medullary rays, in this way this cortex remains extended here. This cortex remain extended here on both the lateral side of the pyramids. This is the medullary rays, or this is the extension of cortex. Extension of cortex here. Here also. This is known as columns, renal columns, or columns of Bartine. This is known as columns of Bartine. The lateral boundaries of the pyramids are the extension of the cortex. They act like anchors or hook, otherwise it will go out. So it anchors like this. These areas are known as columns of Bartine or renal columns. These columns of Bartine have been discovered by Excretory Excretory Bartine, Excretory Joseph Bartine. He is a French anatomist. He was French anatomist. He was the discoverer. Named after the discoverer, this column are known as columns of Bartine. He discovered in 1744. 1744, 1844, 1944. That is almost 200 years. So he has discovered this, and his name was Excretory Joseph Bartine. He was a French anatomist. The papilla here, this is the papilla, renal papilla. These are renal papilla. These are perforated. These are perforated. That is, they are having little hole, and they actually receives the calyx which I am coming. So this is apex or papilla. This is the base of the renal pyramid. These lateral boundaries of the renal pyramids are infested or invaginated by columns of Bartine, named after Excretory Joseph Bartine, French anatomist, in 1744. The renal medulla, here it is darker in color. The renal medulla, it is the site of secretion of prostaglandin, a hormone, prostaglandin, PGA1, PGA2, PGA1 or 2, like that. So these are all prostaglandins is secreted from the medulla portion of the kidney. Clear? Now, since this is the main structure we are concerned, now there are the main unit, functional microscopic units are there, that we are coming, that is, we know that is nephron. Clear? So nephrons are situated here in the cortex. In the cortex, nephrons are situated. These nephrons are the main filtering unit or microscopic unit of kidney, which I am coming, but I will draw the nephron in some other way so as to enable you to understand this. Now, nephron, we know that it consists of this and that. From class 9 onwards, you have started learning that what is nephron. Now each kidney is made up of how many nephrons? 1 million. 1 million. 1 to 1 and more. 1 million nephron in this kidney, and in this kidney there is another million. So you are the owner of 2 million nephrons, not rupees. So 2 million, 2 to 2.5 million nephron you are having in both of your kidney. Now this nephron, we are drawing it in a this way, easy way, and next time I will draw the main nephron, how it looks. First we will write the nephron in this way. Nephron starts with a blind area like a capsule, and in which the blood arteriole enters. Renal, arteriole. From artery, then arteriole, then arterioles. The renal arteriole enters through this area. Now, what happens? Next, it goes inside and it comes out like this. So renal arteriole forms some tuft. This I have made one tuft. This is going here and coming out. It is making one tuft. You can make it another tuft like this. So it forms a tuft of arteriole. Like suppose one arteriole is going, it is making a loop, again loop, again loop, again loop. So many, total 40 to 50 loops it produces. These loops anastomize, that is joins with one another. These loops joins to form this arteriole. This is known as efferent arteriole, and this is afferent afferent arteriole. So we should write that afferent arteriole invaginates, enters. This is known as Bowman's capsule. I am coming. Bowman's capsule, the first part of nephron. This afferent arteriole enters or invaginates in the Bowman's capsule. This is a capsule-like structure where it forms a loop. It forms 50 loops or 10 to 15, 20 loops here itself. So it makes the loops here and it comes out as efferent arteriole. Mind it, it is forming loop, not capillary. Clear? So these loops are like this, Suppose it is going here, it is making a loop. Again, it is making a loop. Again, it is making a loop. Again, it is making a loop. So these loops they are forming capillary tuft. These are the glomerular tuft. So these loops, here from it comes and here from it goes out. So these are loops impregnates the Bowman's capsule. So these loops are the main starting site of filtration of the nephron. Now, whenever it filters out, why it will filter? Why it will go out? The problem is that whenever afferent arteriole enters, it has got a cuff-like muscle. It has cuff-like muscle just in the entry. The afferent arterioles has cuff-like muscles. These are called juxta medulla, juxtaglomerular cells. It is cuff-like. C-U-F-F. Cufflinks, you know, cufflinks. This cuff, the border of the shirt is little bit hard, so it makes a little bit hard to enter. So these are called juxtaglomerular cells or juxtaglomerular cells. It is a cuff-like structure. It enters. Now, why the blood will filter? So there should be some pressure inside. So it is short. This afferent arteriole is broad and short. This bigger one, and it is short in structure. So it is broad and short. This one is efferent arteriole is long and narrow. Long and narrow. Short and oblique. Sorry, the afferent arteriole is short and wide. So it is diameter is more. And this is long and narrow. Diameter is narrow. So I am making this one is short and broad, having the juxtaglomerular cells here. And this is long and narrow. This arrangement has been made that the blood flow should be more. The BP will be more. The pressure will be more. So this arrangement has been made to make the blood flow, at least it is 70 mm of Hg. So this 70 mm of Hg is far more than the other capillary networks elsewhere in our body, because we have artery, then it forms to arterioles, arterioles to smaller arterioles to capillary networks. These capillary networks brings some other blood coming out is venules, venules, venules. So these capillary network do not have this type of 70 mm of Hg. This is less than 70 mm of Hg. This is forming network. This is although not forming network, but the afferent arteriole is forming loop, which anastomize to form the efferent arteriole. This is A, this is E. This arrangement, this is wide and short, short and wide so that it can pump more, and this is short and narrow so that blood pressure will be there so as to enable the waste product or urine from this blood. So it is going out of this blood. Out of this is the main filtering unit. It starts from here. We will come with the Bowman's capsule. Now, nephron, these nephrons are of two types. These nephrons are of two types. Based on the position of the glomerulus, it is divided in the position. Where the glomerulus are here, nephron starts from here, and this is the glomerulus position. If the glomerulus is here, if the glomerulus is here, so based on this position, there are two types of nephron. The corticals, these are called superior glomerulus or superior nephron. That is, glomerulus is superior. So we should write superior glomeruli. And this we will call, this is near the medulla. So this is known as juxta medullary nephron, whose glomerulus is near the medulla. So juxta medullary glomeruli. This is juxtaglomerulary medullary glomeruli, and this is superior glomeruli. It forms 85% of the total number of nephrons present in one kidney, and this constitute 15% of the total So these are the two types of nephrons present in our kidney, and one which is superior in the cortical position. It depends on the presence of glomeruli. If it is in the cortex, it is 85%. During a normal person, whenever a normal person, in case of normal person, 85% of the superior nephron works, and in case of stress situation, this 15% starts working. Otherwise, it remains at rest. So these are the two types of nephron we have come across. Now the nephron consists of, this nephron consists of Bowman's capsule, and here forming loop or glomerular tuft. This is efferent arteriole. This is known as glomerulus. So this is glomerulus. This is afferent arteriole. This is efferent arteriole. This is known as glomerulus. This is known as Bowman's capsule. This is known as Bowman's capsule. Bowman's capsule. The part of the nephron is known as Bowman's capsule. This Bowman's capsule has been discovered by British anatomist histologist, one who discuss with the histology, that is different types of cells and structures and muscles, histology, position, anatomist, histologist, and he was also ophthalmologist. He was surgeon also. And he was ophthalmologist. So he was a British surgeon, he was anatomist, histologist, surgeon and ophthalmologist who deals with microscope. He has discovered, his name was Bowman. His name was Bowman, Sir William Bowman. His name was Sir Bowman. His name was William Bowman. He has discovered this. It consists of 2 layers. We'll come into the detail. This consists of, so nephron will start from, the nephron it starts with this. Along with glomerulus and Bowman's capsule, this glomerulus and Bowman's capsule collectively called renal corpuscle, or it is known as Malpighian corpuscle. So nephron is made up of Bowman's capsule and glomerulus, or you can say nephron is made up of Malpighian corpuscle. Nephron is made up, the first part is known as renal corpuscle. But this contains this Bowman's capsule as well as glomerulus. Then here the filtration starts. It filters the urine. So along with urine, so many other things pass because it has the pressure of 70 mm of Hg. So almost all things other than the cells and big proteins remains inside the blood, and the waste, including the essential items, goes out along with urine. So this is the first filtration urine, and this comes here. This is known as PCT, proximal convoluted tubule. That is, mini tube is known as tubule. So this is known as proximal convoluted tubule, and thereafter this proximal convoluted tubule forms a loop. This loop is known as loop of Henle. We know this. This is known as loop. It is hairpin-like structure. Loop of Henle. Loop of Henle. Who has discovered this? German. Loop of Henle is known as German anatomist. It is known as Friedrich, something like that. Friedrich Gustav Jacob. Friedrich. F-R-I-E-D-R-I-C-H. Friedrich Gustav Jacob Henle. Friedrich Gustav Jacob Henle is the name of the discoverer of loop of Henle. Friedrich Gustav Jacob Henle. He has discovered this loop. So his name, after his name, it is known as loop of Henle. Now this is proximal convoluted tube. This is loop of Henle. Again it becomes convoluted. It is known as distal convoluted tube. Clear? So nephron consists of this. Glomerulus and Bowman's capsule together forms the renal corpuscle, and then proximal convoluted tubule, and then loop of Henle. Loop of Henle is further divided into, this is known as, this is coming here, this is known as descending, D-E-S-C-E-N-D-I-N-D, descending limb. This is loop of Henle. This is known as ascending limb. It is ascending that side. Ascending limb. This ascending limb is made up of two things. This is thin, this is thick. But this is also ascending limb. Of loop of Henle. This is thin, right thin ascending limbs and thick ascending limbs and distal convoluted tubule. And now this distal convoluted tubule will come here, and the efferent arteriole, what it does, will come. First of all, we should know that this is superior nephron. That it is glomerulus situated in the cortex, and this is made up of Bowman's capsule, glomerulus together from this mammalian corpuscle, then PCT, then loop of Henle. This is the descending limb. This is the ascending limb, is divided into two, thin and thick, and then it goes to forms the distal convoluted tubule. And from distal convoluted tubule, it will go to collecting tubule, and the urine will pass out, will come. That will come in the next class. Up to this, we are concluding today. In the next class, we will go into a big detail of this, because so many cells are there which helps for proper filtration and reabsorption. Till then, take rest and please do subscribe and share it. Thank you very much.